Hello everyone. Thank you for joining us for today's joint webinar between Lonza and the Ecological Research and Development Group, ERDG. My name is David Herbst, and I am one of the product managers for our endotoxin detection product line. With me today is Glenn Gaubrey, the founder and director of the ERDG, and to answer any of the regulatory questions that you might have, Alan Bergenson, our regulatory affairs manager, is also here with us. Today we will be discussing the state of the horseshoe crab and the risk to this natural resource, conservation efforts, and how you can move your lab away from an animal-based testing method to PyroGene, our recombinant factor C endotoxin detection assay, a reliable, animal-free alternative to LAL and TAL. Throughout today's presentation, feel free to submit questions using the Questions tab on your screen. Questions will be answered at the end of the presentation, and for those questions that we don't have the opportunity to answer, you will receive a written response within 2 to 3 business days. Everyone who registered for today's presentation will receive a PDF copy by email. The webinar will also be archived on our website for future viewing should you want to revisit any of the topics discussed today. If you should encounter any technical problems, please click on the web link provided in the questions pane of the control panel on your screen. Here you will find the dial-in information for our web host. They will assist you in resolving the technical issue. At the end of the presentation, you will see a feedback form, and we would greatly appreciate it if you would take a few moments to fill this out, as your feedback is very important to us. Today we will be discussing the following topics. First, Glenn will be presenting the increasing demand on the horseshoe crab and the risk to this natural resource. After Glenn is done, I will discuss how you can move your lab away from an animal-based testing method to PyroGene. Go ahead, Glenn.
Hello, everyone. I'm Glenn Gaubrey, and as Dave indicated, I'm the founder and director of the Ecological Research and Development Group, or ERDG. We are a nonprofit organization and the only organization in the world dedicated to the conservation of the world's 4 horseshoe crab species. This statement lies at the core of ERDG's conservation philosophy. I encourage you to visit our website and Facebook page to learn more about our conservation initiatives. However, my purpose today is not to present the work of ERDG, but to engage your mind behind a problem of great significance regarding the conservation of the world's 4 horseshoe crab species, and the role you play through the decisions you make, whether consciously or not. What I will be talking about is the size of the global healthcare industry and how its growth is directly tied to 3 of the world's 4 horseshoe crab species, and whether this growth can be sustained in light of what we know about the health of the world's horseshoe crab species. The healthcare industry, specifically manufacturers and users of endotoxin detection products, are in a unique position to ensure the stability of a natural resource struggling for survival through much of its range, and only marginally stable along the Atlantic coast of the United States, where LAL is derived. Your decisions can have a profound impact on the conservation of this remarkable mariner, and the continued growth of your industry upon which the health of billions of humans and animals depend. First, let's take a minute to look at the life cycle of the horseshoe crab. Although there are many differences between the 4 species in terms of habitats, spawning patterns, and survival rates, their life cycle is pretty much the same. The species is slow to mature. As you can see from the graph on the right, it takes 10 years to reach adulthood, actually 12 years for females. The juvenile will molt each year, growing larger with each successive molt. During these developmental years, the juveniles are predated upon by a host of marine creatures. In fact, only 30 out of about 1 million survive their first year. Once they reach adulthood, they have few predators other than humans, and if not exploited in some manner or another, can be expected to live another 20 years or more. It is only when they become adults that they begin their annual spring migration, from the shelter of deeper waters to their spawning beaches. Females burrow into the sand and lay masses of green eggs, which are then fertilized by males around her. Each female will return to the beach on successive tides, laying as many as 4 to 7 egg clusters with each tide. Each cluster, which incubates 5 to 8 inches below the surface, contains about 4,000 eggs. A female typically lays about 25 egg clusters each year, or around 100,000 eggs. At this time, when they are most vulnerable, they are harvested for either TAL or LAL, as well as bait for several fisheries, human consumption, and folk remedies in certain parts of the world. It is this often unregulated harvesting pressure, coupled with the loss of habitat, that poses the greatest threat to the future survival of these remarkable animals. Unfortunately, the U.S. has its own history of excessive harvesting of horseshoe crabs. The picture on the left shows how horseshoe crabs were stacked and transported for fertilizer and livestock feed during the mid-1800s to the mid-1900s. Harvesting for this purpose dropped throughout the 1950s and ceased altogether in the 1960s due primarily to the industry's shift to chemical fertilizers. Unfortunately, just as the horseshoe crab population began showing signs of recovery, the commercial harvesting of horseshoe crabs for bait in the eel and whelk fisheries began in the mid-1990s. In 1999, ERDG launched an initiative to reduce the demand for horseshoe crabs as bait in the U.S. whelk and eel fisheries. Through a partnership between ERDG and a local whelk fisherman who had devised a bait-saving device, ERDG manufactured and distributed over 15,000 of these devices to whelk and eel fishermen up and down the Atlantic coast of the United States, which was ultimately responsible for a 25% reduction in the number of horseshoe crabs needed for bait in these fisheries. ERDG is currently working with our Mexican partners in the development of an alternative bait. If successful, it could eliminate the need for horseshoe crabs as bait altogether. This GEAR initiative, coupled with regulations put in place by the Atlantic States Marine Fisheries Commission, or ASMFC, and other conservation measures, has reduced the annual coastwide harvest of horseshoe crabs in the U.S. fisheries, bait fisheries in particular, to under 1 million coastwide, from a high of over 3 million. This has resulted in a stable but increasing horseshoe crab population in many of the mid-Atlantic states. However, the population is still declining in the New England states. ERDG is presently partnering with the endotoxin detection industry with the belief that this industry and their customers have the potential to make a significant difference in the conservation of the world's 4 horseshoe crab species in ways not possible through traditional conservation strategies. As this slide suggests, I'm inviting you to take the big picture as it pertains to the world's horseshoe crab species and the endotoxin detection industry and its customers. It is questionable whether current harvesting levels for TAL and LAL can be sustained, much less meet the future demands of this rapidly growing market. For unlike the whelk and eel fisheries in the United States that only benefit those who enjoy such delicacies, the TAL and LAL industry is uniquely responsible for the safety of much of the world's pharmaceutical and medical devices, which impacts the lives of every man, woman, child, and domestic animal in the world who are dependent upon medical services for their health. A few of us truly understand how deeply TAL and LAL industry affects the lives and the lives of those around us. As I just said a moment ago, that every man, woman, child, and domestic animal in the world who are dependent upon medical services for their health rely on TAL or LAL. This is because the safety of much of the world's pharmaceuticals and medical devices are required to be tested for the presence of life-threatening endotoxins prior to public use. And the most reliable endotoxin detection test available is made from the blood of the horseshoe crab, either Limulus or Tachypleus. This involves 3 of the world's 4 horseshoe crab species. To put this in visual perspective, this slide shows just a few of the product categories dependent upon TAL and LAL for endotoxin detection. I am confident that each one of us are connected to the blood of the horseshoe crab through one or more of these product categories. There is no indication that the world's human and animal population will become less dependent upon medical services in the years to come. In fact, as our global population expands, ages, and medical advances improve and/or prolong life, we will become more, not less, reliant upon endotoxin detection methodologies, which means TAL or LAL. So let's take a look at the main drivers behind the global increase in the demand for endotoxin detection. A portion, around 25% of the medical device market, is dependent upon TAL, LAL for endotoxin detection. Although the medical device market is about half the size of the world's pharmaceutical market, approximately $350 billion annually, it is showing signs of rapid growth, driven in part to greater life expectancy and aging population. Currently, the medical device market is dominated by U.S. companies, making up approximately 45% of the total revenue, with Europe coming in second at approximately 25%. However, as this slide indicates, the Asian market is rapidly growing, and currently a large percentage of endotoxin detection in China and India is dependent upon TAL, which relies on two species of horseshoe crabs that are in serious decline. Now let's take a look at vaccines. Although vaccines are a small segment of the global pharmaceutical market, they represent the fastest growing segment of this industry and are heavily reliant upon endotoxin detection. According to the World Health Organization, the vaccine market projected growth over the next 15 years is 10 to 15% annually versus 5 to 7% for pharmaceuticals. And these figures do not include the rapidly growing veterinarian medicine market. Much of this growth will occur in emerging markets in countries such as China, India, and Brazil. Again, our concern should be on the countries that are heavily reliant on TAL for endotoxin detection and the effects of diminishing Asian horseshoe crab population could have on the projected growth of this segment of the pharmaceutical industry. Of course, keep in mind I am speaking to you as a conservationist, so my concern is the survival of the species, not whether or not the pharmaceutical industry meets projected growth. However, there is no reason why these two objectives cannot coexist. As you look at this slide, pay attention to the section labeled Rest of the World, for this represents the emerging markets. Countries such as China, India, Brazil, that I mentioned before, will be major global players in vaccine research and development. These countries are responsible for producing more than 86% of the vaccines by volume and projected to grow to 87% by 2020. China, being the world's largest vaccine producer, produces more than 1 billion vaccine doses annually and is prepared to take on a larger role in the international vaccine market. With the world's largest population and 17 million newborn babies each year, this should come as no surprise. If we just consider the global use of vaccines alone, we can see how reliant we are on horseshoe crabs via the endotoxin detection industry. Okay, now let's take a look at the pharmaceutical market as a whole. It is important to keep in mind that not all pharmaceutical products are tested with TAL or LAL. However, it is safe to assume that if it comes in contact with your blood, lymphatic system, spinal fluids, or mucous membranes, it is tested for the presence of endotoxins. In essence, that which does not pass through the body via an oral pathway. This is a considerable share of the market. However, what is most important about this slide is the overall projected growth from 2006 to 2016, as well as the growth of the emerging markets from 14% to 30%. Here again, these emerging markets represent many of the same Asian countries who are experiencing rapid growth in the vaccine market. This slide drills a little deeper into why these emerging markets are experiencing such rapid growth. An emerging market is defined as the percentage of growth into the 5K or above income bracket. These individuals now have access to medical services previously unavailable to them. As you can see, China's income growth by this matrix dwarfs all other emerging markets combined. There is also a growing trend to outsource product manufacturing to these emerging markets, which may make good use economically if it were not for the fact that a good portion of this emerging market is dependent upon the blood of two horseshoe crab species that are in serious decline. Although it is true that not all products in the charts and figures I have presented utilize endotoxin detection, it is also true that the same charts and figures fail to include the amount of endotoxin detection utilized in the development of new products. This graph is a visual reminder of the amount of R&D and the time associated with bringing products to market. This highlighted box shows the scope of endotoxin detection utilized during the R&D process. For every 250 drugs that make it to preclinical testing, only one may make it to FDA approval. Unfortunately, I'm unable to give you data on the size of the TAL and LAL industry itself in terms of annual revenue. However, the size of the TAL and the LL market is not really important, but rather the size of the market they serve. For it is the growth on the consumer side that places pressure on horseshoe crab resource needed for endotoxin detection. Question: can the world's horseshoe crab population meet the growing demand of the global healthcare industry? Although we lack definitive data on the size and the health of the Asian horseshoe crab population, by all accounts it is diminishing. Yet, as I just indicated, the Asian healthcare industry is growing at an unprecedented level and is largely dependent upon TAL, or the Tachypleus species, for endotoxin detection. In the United States, home to Limulus, harvest of the horseshoe crab for the LAL industry is regulated. With the United States horseshoe crab population just beginning to show signs of recovery after years of decline, it is questionable whether the U.S. horseshoe crab population can manage the growth of the LAL consumer market, much less the TAL consumer market, if there were to be an issue of supply and demand. Is it wise to have the global healthcare industry so directly tied to a diminishing and/or marginally stable resource? This map shows the distribution of the world's 4 horseshoe crab species. Not surprisingly, the world's 4 LAL producers are located along the east coast of the United States, and the world's 4 TAL manufacturers are located along the west coast of China. For Limulus, we are just beginning to understand the size and health of the population. However, as a result of aggressive management policies, we are seeing signs of recovery throughout most of its range. The challenge for the 3 Asian species is much more complex. For not only is its range extensive, it encompasses a host of countries and very different social, economic, and environmental priorities. We have considerable amount of information about the species in areas where the population has declined, but much less information about the species in areas where the population remains strong. It is in these areas of relative health that are of major concern. For as the need for TAL grows and the resource in traditional harvesting areas decline, these healthy, often unregulated populations will be the first to be exploited, if they're not being exploited already. As a result of the serious decline in the horseshoe crab population during the 1990s, the Atlantic States Marine Fisheries Commission, or ASMFC, a governmental regulatory body represented by each of the states along the east coast of the United States, implemented an Interstate Fisheries Management Plan, or FMP. This plan focuses on the stability of the species and has been revised 7 times since its inception to keep pace with changing conditions in the horseshoe crab population. It is the important point to take away from this slide is the biomedical exemption based on bleed and release mortality of 57,500 horseshoe crabs, which is calculated at 15% of the total biomedical harvest. The LAL producers are currently at the upper limits of their harvesting allocation based on the industry average mortality of 15%. In fact, every year since 2007, they have exceeded the mortality threshold set down by the AFMSC. A review process is underway to determine what, if anything, should be done. Unless this threshold is raised and/or the level of mortality drops, there is not much room for growth, certainly not at the levels projected by the healthcare industry, who are dependent upon endotoxin detection. With a bleed and release policy, LAL producers have been able to manage growth through improved harvesting, handling and release protocols, as well as advancements in manufacturing and testing methodologies. However, with production waste at a minimum and testing methodologies already fine-tuned, LAL producers will need to harvest more animals in order to keep pace with global demand. So now let's take a look at the 2 Asian horseshoe crab species that TAL is derived from. TAL is only produced in China and is primarily used in China, some in Russia, very little in India, and hardly any in Japan, South Korea, and Southeast Asia. Chinese multinational pharmaceutical and medical device companies that manufacture in China, yet sell worldwide, purchase and test their end products with LAL. However, with the world's largest population of over 1.3 billion people and a fifth of the world's population, and the largest aging population in the world, China by itself is a sizable healthcare market. Unfortunately, we do not have reliable harvesting figures for China's TAL industry, nor knowledge of how it may be regulated. We are only just beginning to gather data on where, outside of China, Tachypleus is harvested for TAL. What we do know is that Tachypleus is generally harvested in pairs and bled to death, then sold to a secondary market, resulting in 100% mortality. The limiting harvesting numbers that we do have suggest a drop in availability from 600,000 pairs 5 to 10 years ago to less than 100,000 pairs currently. With the 3 Asian horseshoe crab species trending towards decline, how will TAL producers keep pace with their country's growing healthcare market? I think by now you can see where this is going. The demand for horseshoe crabs throughout its spawning range is placing significant stress on the species and hindering conservation efforts, which in some parts of the world face considerable challenges. Building on the global concern for the survival of the world's 4 horseshoe crab species and collective research presented during the last 2 international workshops on the science and conservation of horseshoe crabs, the IUCN Horseshoe Crab Specialist Group was established. Outside of the United States and throughout much of the developing world, the International Union for the Conservation of Nature, the IUCN, is a respected body for the assessment of species-specific data. I sit on the steering committee for this newly formed Horseshoe Crab Specialist Group, and I chair the Industry, Trade, and Use subgroups, which has members from 2 of the LAL producers. The primary purpose for this specialist group is to assess and mitigate the risks to the sustainability of the world's 4 horseshoe crab species. Unfortunately, we do not yet have sound data on the health of the 3 Asian horseshoe crab species. However, we do know is that the horseshoe crab population in Japan, Taiwan, China, Hong Kong, and Singapore, once vibrant, are now seriously depressed and endangered. At the conclusion of the 2nd International Workshop on the Science and Conservation of Horseshoe Crabs held in Hong Kong in 2011, a vote was taken on various action items presented by each of the various work groups, which were then distilled down to the most important. These are the 6 that rose to the top. As I already indicated in the last slide, we have completed item number 1, and by talking to you today, we're working on item number 3. Without a regulatory process on TAL, similar to what is in place for LAL, the current rate of harvest for Tachypleus is unsustainable. Failure to address this problem will continue to add to the decline of the 2 Asian horseshoe crab species and shift the pressure on the U.S. LAL market to absorb the difference. As a result of sound U.S. management policies implemented since 1999, Limulus is beginning to recover. However, some of its range are still depressed. It is questionable whether the LAL industry can offset a decline in TAL availability in a rapidly growing healthcare market. Clearly, in addition to the regulatory process and the implementation of harvesting and husbandry best practices, we also need to invest in the development and utilization of endotoxin methodologies that reduce our reliance on horseshoe crabs and/or replace them altogether. For the sustainability of the endotoxin detection industry and the sustainability of their customers' supply line will ultimately require such a transition. The question is whether the pharma industry will drive this transition and by doing so become partners in the conservation of this remarkable animal, or wait until it's too late to make a significant difference. There are some solutions to this problem, and David, our next speaker, will be presenting one of them. I have merely outlined the problem, and hopefully in doing so, engage your support behind the conservation of this remarkable animal. I would like to re-emphasize that it is the size of the consumer market, in this case the global healthcare market, that places pressure on the horseshoe crab resource needed for endotoxin detection. In the coming months, ERDG will launch a new section on our website designed to more effectively delineate the problem we discussed today and hopefully provide you some tools to have a meaningful conversation with others about how your industry can become a driving force behind the conservation of the world's 4 horseshoe crab species. In the meantime, please visit our website and follow us on Facebook. I thank you very much for your time today, and I look forward to answering your questions a little later. Dave, I'll turn it over to you.
Thank you, Glenn. Now we will have Now we will look at how users can decrease the demand on the horseshoe crab and begin to move their product testing to an animal-free alternative to the LAL and TAL-based assays. I am sure by now you all know why there is a need for an alternative method, but I would like to summarize where this need is coming from before we continue. The LAL assay is one of the few assays which still relies on an animal source for a key raw ingredient. Here is our concern. On one hand, the number of tests performed each year continues to grow, and on the other hand, the fishing quotas remain restricted while this demand increases. QC laboratories must face the risk that there won't be enough raw material, LAL and TAL, available to fully meet the global demand. With the TAL supply in decline and the demand growing, if an alternative is not considered, the demand on LAL will increase dramatically. This will either lead to a higher price of the test or result in shortages of a critical in-process and end-process release test. In addition to the obvious driver of avoiding shortages, moving to PyroGene also allows companies to meet initiatives to move away from animal origin materials and would allow companies to meet specific European initiatives to reduce animal use in testing. Lonza's animal-free solution to this challenge is the PyroGene recombinant factor C assay. It is currently classified as an alternative method, but when you look at it, it's a photometric assay remarkably similar to the classic methods like our LAL-based assays. So how does PyroGene work? PyroGene utilizes a recombinant form of the factor C enzyme to detect endotoxin. This is the same endotoxin binding enzyme that is the first step in the classic LAL assays. However, instead of having factor C activate a cascade of other enzymes, it directly acts on the substrate, allowing the instrument to detect the change in light intensity. The PyroGene substrate that is acted upon has a fluorescent tag, which gives a much wider dynamic range with better resolution than the classic LAL methods. The fact that we use a single recombinant protein only and do not need the other proteins of the LAL cascade has several benefits to the user. Let's look closer at what I have just described. On this slide, you can see a comparison of the LAL cascade in the left graphic alongside the recombinant factor C activation pathway on the right. At the very top of both images, you can see endotoxin activating the first enzyme in the cascade, factor C. In the case of, going further down the cascade of the LAL-TL cascade, factor C in turn activates factor B, which in turn activates the pro-clotting enzyme. In the case of gel clot and the kinetic turbidimetric assay, the activated clotting enzyme cuts coagulagen to form coagulin, which allows a measurement of endotoxin present. In the case of the chromogenic assays, a substrate is cleaved, which allows measurement of endotoxin based on the intensity of yellow color in solution. Please also note in the middle of the picture that the cascade can also be activated by glucans. This second pathway makes LAL and TAL-based methods sensitive towards glucans. Now let's look at the picture on the right side of the slide. The graphic shows the reaction for the PyroGene assay. Once again, you have endotoxin activating factor C. The activated recombinant factor C then does not activate a cascade, but directly acts on a fluorogenic substrate, allowing measurement of endotoxin through a fluorescent signal. You'll notice that the glucan-activated pathway is not present here, meaning the PyroGene assay is not sensitive towards glucans, one of the benefits of this assay. This slide shows a further comparison of how parts of the cascade have been replaced by other methods for amplification and detection throughout the development of the endotoxin detection assays. On the left, you see a green arrow which indicates the development timeline up to today. In the middle of the slide, you have the signal amplification step, and on the right, the method of measurement. The first assay, established in the 1970s, was the gel clot assay, which was based on a naturally occurring reaction in the horseshoe crab blood. Endotoxin was detected by observing whether or not a clot formed when inverting a glass tube. The gel clot assay is, however, only a semi-quantitative test. The same cascade was used in the next development step, the turbidimetric assay, which was the first quantitative assay. In this assay, the turbidity of the beginning clot was used to quantify endotoxin levels. The chromogenic assays went a step further and used a chromogenic substrate as the last step in the cascade. Instead of measuring turbidity, now the development of a yellow color was measured. The most recent development is the recombinant factor C assay, which can be seen at the bottom. Here the cascade is truncated even further and replaced by a single step, which gives a fluorescent signal. Again, I want to point out in the blue box that in each case it's recombinant factor C, which is detecting endotoxin. While the assay itself is remarkably similar to the quantitative LAL methods, there are several benefits that PyroGene provides. First, since it utilizes a recombinant enzyme, we have a very good batch-to-batch consistency relative to an animal-derived assay. Again, the assay is endotoxin specific. If you remember the previous slides, there is no secondary pathway for activation by glucans. The assay is statistically more robust, and because recombinant factor C is a recombinant protein, it provides a greater security of supply and avoids the use of animals in testing and reagent manufacturing. The fact that it is a liquid test also makes it easier to use, since you do not have to reconstitute a lyophilized product. Last, it does go down to 0.005 EUs per mil, which is suitable for most samples being tested for endotoxin. To demonstrate the batch-to-batch consistency, we generated standard curves for 8 different lots of recombinant factor C. This is shown here with the 8 overlaid curves. You can see we get excellent reproducibility. While the previous slide showed standard curves down to a sensitivity of 0.01, PyroGene assay now provides sensitivity down to 0.005 as I indicated earlier, and here's a standard curve indicating that level of sensitivity. So how is the PyroGene assay set up? You will note that it is actually, as we mentioned before, is actually very similar to other quantitative methods. It is a single-step endpoint fluorogenic assay, which only takes 1 hour at 37 degrees Celsius. This temperature is the same temperature used to incubate the LAL assays as well. This assay follows the bacterial endotoxin testing methodology as well, and we'll come back to this point a little later. PyroGene uses liquid rather than lyophilized reagents, as we mentioned before. So the assay is set up mixing these liquids. The assay is performed in a standard 96-well microplate using an incubating fluorescence plate reader, which is controlled by our endotoxin detection software, WinKQCL. As with the LAL-based assays, this software also does the calculations and provides the assay reports. The only difference is that the reagent is not manufactured from lysate, sourced from horseshoe crab blood. So now let's talk about the validation of recombinant factor C as an alternative method. To validate as an alternative method, Lonza actually performed this series of tests as well. We performed the following tests according to the requirements outlined in USP Chapter 1225. Using a multi-center study located in both the US and Europe, we compared kinetic QCL with the PyroGene assay. We tested 10 different pharmaceuticals, with 9 out of 10 of these being blind tests. The products had varying release limits. They were tested at a maximum valid dilution, and then at half MVD, and then at 1/10 MVD. The labs were provided with already pre-diluted samples, and all assays were performed according to the package inserts. Standard curves were used that range from 0.01 to 10 EUs per mil, and the PPC spike was 0.1 EUs per mil. The samples tested were water for injection, lactated Ringer's injection USP, 0.9% sodium chloride injection, 50% dextrose injection, human albumin, gentamicin sulfate injection, recombinant insulin, vancomycin, erythropoietin, and hemodialysate. The evaluation criteria for the validation studies are outlined in USP Chapter 1225. It indicates a need for specificity, precision, accuracy, linearity, and says that the detection and quantification limit as well as range and robustness must be at least comparable to the compendial methods. The results of the evaluation were published in the Pharmacopeal Forum, Volume 36, Issue 1, from January-February 2010. The following slides will summarize some of the data included in this article for Stimulus. So the first point, the need for specificity, was shown by comparing the two reagents. And here we compared the two reagents' reactivity to glucans. The purple bar is the result of the test with PyroGene, and the green bar, the test with the kinetic QCl chromogenic assay. On the left side, you can see a tall green bar next to a small purple bar. This was a sample that contained glucans, which showed a clear response with their chromogenic assay. In comparison to that, PyroGene was not activated. On the right side, another sample was tested with glucan blocker. There is nearly no reaction anymore for the chromogenic method, showing that the previous reaction was indeed caused by glucans. This experiment showed that PyroGene is indeed endotoxin specific, showing no response to glucans at all. The second point called for its precision. Defined in USP 1225 as the degree of agreement of individual test results when the method is applied repeatedly to multiple samplings of a homogeneous sample. The precision of an analytical method is usually expressed as the standard deviation or relative standard deviation or coefficient of variation, in this case, of a series of measurements. Precision was determined in the multi-center study as seen in this slide. The top graphic shows recombinant factor C tests conducted at 3 locations. Each was provided with 3 samples of different endotoxin concentration. The low sample was approximately 0.03 EUs per mil. The medium sample was 0.3 EUs per mil, and a high sample at 3 EUs per mil. 18 tests were performed per location, making a total of 54 tests. The coefficient of variation was calculated for the individual locations as well as the overall CV. The lower part of the table shows the results of the same samples tested with the chromogenic assay. If you compare the results over location, you can see that the CVs for PyroGene were comparable to the CVs for the chromogenic assay. With the PyroGene results coming in at 16%, 17% for the medium, and 11% for the high, compared to 19%, 20%, and 16% for the chromogenic assay respectively. We can see that PyroGene is equivalent to the kinetic chromogenic assay with regards to precision. The third demand for alternative methods is accuracy. This was tested in different ways. We compared the recovery of a 0.1 EU per milliliter spike in water for 3 different methods. As you can see from the results, they all showed good accuracy. The endotoxin content found was 0.092 EUs per mil with the chromogenic method, 0.112 EUs per mil with the turbidimetric method, and 0.05 EUs per mil with recombinant factor C. The standard deviation was also comparable, with a very low 0.009 for chromogenic and 0.012 for recombinant factor C. Of course, you wouldn't just test water samples, so we determined accuracy also by spiking multiple products. you can see the spike recoveries shown here. On the left side, we applied the recovery when tested with the chromogenic assay. On the right side, the same samples tested with recombinant factor C. We would accept a PPC recovery between 50 to 200%, which is the limit given by the pharmacopoeia. As you can see, both methods yield spike recoveries which are at least equivalent. Results for recombinant factor C are actually even a bit tighter. We once again used the results of the same 3 samples I showed you earlier. For accuracy is defined as the closeness of test results to the true value. We determined the percent of the results within 25% of the known concentration. You can see the results here. If you compare the 2 assays, you can see that the accuracy of PyroGene is at least comparable, even better than for the compendial method. For the first sample, the one with the lowest concentration, 81.5% of the results obtained with RFC were within 25% of the true value. For the chromogenic assay, this was 74.1%. Likewise, we had a medium sample come back as 44.4% with RFC and 29.6% for the chromogenic assay. And for the high sample, 92.6% for recombinant factor C and 87% for the chromogenic assay. To demonstrate the 4th requirement, linearity, we made a linear regression statistical summary of standard curves from our multi-site study. We looked at the 3 characteristics of the standard curve as given in the left column: the correlation, the slope, and the y-intercept. You can see in the 3rd column that the CVs for all characteristics are very tight, with 0.1%, 1.9%, and 1.1% respectively. This shows that we get a very linear response, and recombinant factor C is a very reproducible method. From our extensive validation studies, we concluded that PyroGene is a suitable assay for most of the products that we tested and can be used in place of any compendial method. However, it will not address interference problems which are due to endotoxin availability. For example, if you have samples which contain proteins that bind endotoxin, or if you have aggregation problems, PyroGene may not solve these issues either. Therefore, normal validation procedures should be followed as with any method, meaning, for example, to do an inhibition enhancement study for each new sample being tested. All in all, PyroGene provides a sustainable addition to or replacement to the LAL and TAL assays. While we could devote an entire hour to the topic of regulatory considerations, let me just touch on a few highlights. First, the new FDA guidance document which was published in June of 2012. This does answer the question, may a firm use alternative assays to those in the USP for compendial article? The answer from the FDA was yes. And as we have discussed earlier, this method should be validated per USP Chapter 1225 and should be shown to achieve equivalent or better results. The FDA specifically mentions 2 examples, one of them being the recombinant factor C assay. The FDA also addressed the question of what is the best process for transitioning from one alternative BET method to another. Here they indicated the following: comparing the 2 tests to verify the equivalence of the new method per USP 1225. For drug, animal, drug, and biological products, the transition to a new method should be submitted in a prior approval supplement, or PAS, or a comparability protocol, or CP, before using the new method for release. For devices, a 30-day notice may be appropriate for changes to quality control testing for all BET testing. In addition to the FDA questions, customers often ask about the validation protocol and timeline. A possible validation scheme is outlined in this slide. One validation can be accomplished in as little as 5 days, assuming that the product has been previously validated with a quantitative LAL method. The validation schema in this slide is identical to that which would be needed for any LAL-based method with the addition of just one step, the validation of alternative method. Lonza offers a full validation protocol that can be followed for your convenience. For further information, please contact scientific support or your local sales representative and we can provide this protocol for you. In summary, we have shown that there are risks to the source of LAL and TAL. An alternative method, PyroGene, is commercially available. This method is sustainable and does not rely on the horseshoe crab. The alternative method has been shown to be comparable to other photometric methods listed in the chapter on bacterial endotoxin testing. Lonza is willing to partner with the industry to facilitate the adoption of the alternative PyroGene recombinant factor C assay. Now that we are done with the presentation, I want to thank you for your attention. And I would actually like to hand it over to Glenn for a few more comments.
Hi. I'd just like to remind you that the conservation of the world's 4 horseshoe crab species is a shared responsibility. Each of us have a role to play, and as you become more mindful of the effects your purchasing decisions can have on a natural resource your industry's growth is dependent upon, we become global partners in the conservation of a remarkable species. So if you have any questions about horseshoe crabs and or how your company can get involved in ERDG's conservation initiatives, please reach out to me or us at www.horseshoecrab.org, easy to remember. And I'd just like to thank you again for the honor and having the opportunity to speak with you today.
We are now going to pause to answer questions. And while we put you on mute, or put us on mute, please take a minute to fill out our feedback form. Please do not post any of the questions in the comment field of the feedback form. Use the questions tab on your control panel. While you fill out that form, our microphone will be on mute for a couple of minutes while we review your questions. We'll be right back. Okay, I'm going to start with our first 2 questions. First, and I'll let Alan answer this, can we test cell culture medium with this method?
Thanks, David. Actually, yes, you can, absolutely. The test would be no different than testing any other pharmaceutical product. The only difference you're going to have there is that there will be no compendial endotoxin limit. You'll have to develop one yourself or determine one yourself based on the use of the product.
Okay, the next question I have is it important to use a black plate for the reading of the recombinant factor C assay? And for this, I would actually say not necessarily, and actually no. If you are using the instrument that we provide, we actually prefer to use a bottom read through a clear plate, and the assay works just fine with that. So essentially you can use the same plate that you're currently using if you're using Lonza provided plates with your kinetic chromogenic assay or turbidimetric assay. One moment while we continue to look at incoming questions. Okay, we have another question that came in. Is the PyroGene assay going to be in the harmonized USP, JP, EP in the near future?
And the answer to that question is right now we are working on inclusion. We've shown the compendia that the assay really is no different than the other technologies that are currently in the BET assays. So we're hopeful.
Okay, and I've got a couple of questions from yesterday that came up, so I want to share those as well with the audience. The first one was, does the FDA regulate PyroGene? I'll let Alan answer that one as well.
Okay, and the short answer to that is no. The longer answer to that is you have to think about why LAL is regulated. LAL is regulated because it is derived from blood of an animal species. So it's not regulated for what it does. It is regulated from where it comes from. And that comes directly out of the Section 356 of the Public Health Service Act in the United States. So Lonza, several years ago, when we developed PyroGene, we wanted to know what FDA agency would regulate PyroGene, and since it didn't come from the blood of an animal, and we sent in a request for designation to the Ombudsman, and it was circulated to both CDRH and CBER, and it came back that because it was not derived from the blood of an animal, and that it was not used to diagnose, treat, or mitigate a disease condition, that it wasn't a medical device. Therefore, no licensing was necessary. It's not that it couldn't meet the criteria for licensure. It could. It's just that from a regulatory standpoint, it didn't need to be.
Thank you, Alan. One moment while we look at some more questions from yesterday to review. Okay, another question was, does the FDA have a PyroGene system?
And the answer to that is yes. The FDA does have several PyroGene systems at their facilities. Now, you have to remember that FDA does not manufacture drugs or medical devices, but they do have to review all of the data that comes in. So the FDA has systems to familiarize themselves with how the assay works.
Okay, and then the last question I'll answer, are we offering opportunities for evaluation? And the answer is absolutely yes. Again, please contact scientific support or your sales representative if you would like to partner with us in evaluating your product with PyroGene. Looks like that's all the time we have today. I really appreciate everybody's time today in attending this webinar, and we look forward to reviewing your feedback. Thank you very much, and have a